Hi, welcome to Learning and Memory, Chapter 2. My background is social psychology, not neuroscience. So in your notes, I've compiled some outside information and resources that provide a more sophisticated and technical overview of neurons in the broader central nervous system. In this video, I'll discuss some more general big picture overview. In this course, we'll focus primarily on the central nervous system and only briefly discuss the peripheral nervous system. Most processes involved in learning and memory are carried out in the central nervous system. To understand the system at a cellular level, we must examine the structure and functions of neurons. There is some variety in the types and structures of neurons, but we'll stick with our common generic neuron. The central nervous system is composed of approximately 86 billion neurons, but it's the connections between neurons that are important for understanding sensory perception, voluntary movement, learning, and memory. By neural connection, we just mean that two or more neurons are communicating with or influencing one another. With connected neurons, we have the presynaptic and the postsynaptic neurons. Presynaptic means before the synapse, and postsynaptic means after the synapse. The synapse is a small space that separates two neurons. This means that when two neurons are connected, they aren't typically physically touching. Instead, they communicate via chemical messages across the synapse. The presynaptic neuron receives information from other neurons or sensory receptors via the dendrites. Dendrites are the branch-like structures that connect to the cell body. The dendrites receive information. The cell body is where these messages that may come from a variety of different neurons are integrated. Some of these messages are excitatory and some are inhibitory. Excitatory messages increase the likelihood that a neuron will fire and communicate with the postsynaptic neuron. The inhibitory messages decrease the likelihood that a neuron fires and communicates with the postsynaptic neuron. If the cell body receives enough excitatory messages to reach its threshold, the neuron will fire an electrical impulse that travels down the axon to the terminal. The electrical signal signals the terminal to release sacs or vesicles filled with neurotransmitters into the synapse. These neurotransmitters are chemical signals that travel across the synapse and bind to receptor sites on the dendrites of the postsynaptic neuron. If the neurotransmitter binds to the receptor site, then that message is integrated in the cell body of that postsynaptic neuron. So we see two types of communication, electrical and chemical. Electrical communication occurs within the neuron when the cell body tells the terminal to release the neurotransmitters. We see chemical communication that occurs between the two neurons across the synapse. It is important to understand communication between neurons because changes in communication patterns may be the physical manifestation of learning. According to the theory of Hebbian learning, when two neurons repeatedly communicate, the connection between them strengthens. Neurons that fire together wire together. Through experience, the communication between neurons is altered. So if through experience, two neurons are repeatedly co-activated, this may lead to structural changes, such as the addition of synapses between the neurons, or the addition of receptor sites in that postsynaptic neuron. These types of changes make the postsynaptic neuron more sensitive to the messages of the presynaptic neuron. These structural changes are called long-term potentiation. More receptor sites and synapses equals more potential for the neurotransmitter to bind to that postsynaptic neuron. Alternatively, if two neurons don't fire together, the connection between them may weaken, resulting in what's called long-term depression. Receptor sites may be pruned, making the postsynaptic neuron less sensitive to the messages of the presynaptic neuron. This suggests that learning and memories are not located within individual neurons, but rather the connection between neurons.